Okay, so what we are going to do today is, first of all, you need to answer this question for your attendance. So this is for Tuesday, April 5th. So make sure that you're finding this on Canvas. It should be in the week 13 module. So what I need you to do is either write me a multiple choice or a fill in the blank question that is concerning what we talked about last Thursday, which was March 31st. And so, and we talked about mitosis and asexual reproduction. So turn that in on Canvas, make sure that you include the correct answer. And just as a reminder, our third exam is scheduled for April 14th. And so as I've been collecting these, I've been putting them into a Google document, which I will share with you on Thursday. So I'll let you know when that is available to you and where you can find it. Okay, so make sure and do that for me. So now what we're going to do is we're going to shift our focus to talk about sexual reproduction, which is accomplished through the process of meiosis. So meiosis and mitosis are similar in some respects and very different in some respects. So we're going to be looking for Venn diagrams in your future. Okay, so just a little bit of a review. So last time we talked about asexual reproduction. Prokaryotes do binary fission, whereas eukaryotes tend to do, when they asexually reproduce, they use budding, fragmentation, and they can also, so this process is accomplished through mitosis in eukaryotes. So remember that prokaryotes don't do mitosis. They don't have a true nucleus. And so either way, if you do binary fission or mitosis, you're going to end up with genetically identical offspring to each other and to the parent. Okay, and so remember we had interphase, where all of our cell was just doing regular cell stuff. It wasn't necessarily preparing to reproduce. And so we talked about there's 3 different phases of interphase. There's the growth 1 phase, G1, the DNA synthesis, we call that S, and then there was the growth 2 phase, which we call G2. And all of that stuff is happening in interphase, where the cell is just kind of doing normal cell stuff. It replicates its DNA and then it has to recover before it does the mitotic division. And so for mitosis, even though this is a very small portion of the cell's life, we talk about it a lot because there's a lot of steps involved. So what happens first is we have prophase. Okay, so this is prophase, where we have our DNA is pairing up. We are seeing the nuclear envelope is falling apart. We see spindle fibers in animals starting to arrive on the scene. And then we go to metaphase, where we have our chromosomes lining up and we have our spindle fibers attaching to each of the sister chromatids. And then in anaphase, those sister chromatids are pulled apart. Telophase, we see the nuclear envelopes of our 2 new nuclei starting to reform. And we also have that cytokinesis that we can see here. And so for plants, that involves a cell plate. And for animal cells, it involves the cleavage furrow. Okay. So now let's talk about sexual reproduction. So, okay, so again, just a little bit of a review. Our asexual reproduction occurs in our somatic cells, or our body cells. Sorry about that. For most animals, when they're in their adult phase, like we are in our diploid phase. Our multicellular state is as diploid, so we have 2 copies of each chromosome in all of our cells. Through asexual reproduction, we get daughter cells that are genetically identical, and we do this through mitosis. Sexual reproduction, on the other hand, that only happens in eukaryotes. That's an important point. This occurs in our germline, or our sex cells. So we usually call those gametes. So depending on what kinds of gonads you have, that's going to determine what kinds of gametes you make, whether that's eggs or sperm. So these are going to be haploid. cells, they have half the number of chromosomes. And so in our case, and us animals, that means that they are 1N. Okay, so these daughter cells are genetically distinct from each other and the parent cell. You're going to hear me say that a lot, so maybe that's something that you want to know. And then, of course, sexual reproduction requires meiosis. Okay. So when we talk about sexual life cycles, and you'll go more in depth in these when you take 182, but basically what we're doing is we're alternating between a haploid and a diploid stage. And where the multicellular individual is, it depends on what kind of critter you are. For us animals, we have our a life cycle like this, where we have a diploid zygote that undergoes mitosis to make a multicellular diploid individual. We have meiosis is occurring. It's going to make special cells that are going to become sex cells. Those sex cells will find each other, egg and sperm meet, and then fertilization happens, and now we're back to being diploid. And so it doesn't matter the complexity or the type of life cycle that you have, the things that they have in common are, we've got meiosis is the gate to get from being diploid into being haploid. We have fertilization, which is the gate to get from being haploid to being diploid. We always see a diploid zygote when we have the fusion of our gametes. So zygote gametes are also things that you would say if I said, okay, these doesn't matter what kind of life cycle you have, what are the things that you have? And you would list off meiosis, fertilization, gametes, zygote. Okay, 2 processes, 2 types of cells. So in plants, we see this weird thing. We have the sporic meiosis, or an alternation of generation. Again, you'll cover these details when you get to 182, but just to kind of plant this in your mind, because this is one of those weird things that plants do that a lot of students have a hard time with. So if you pick your favorite plant, let's say a saguaro cactus, because here we are in Arizona, why not? So that is a diploid multicellular individual, has specialized cells that undergo meiosis to make haploid spores. Those spores are going to undergo meiosis. Nope, sorry, let me say that again. The spores are going to undergo mitosis to make multicellular individuals. Those multicellular individuals are going to have specialized cells that make gametes. Gametes are going to find each other. Fertilization is going to make a zygote. So boom. So there's some details in there, but there we got our meiosis and fertilization. We got gametes and zygote. Okay. In fungi, they have zygotic meiosis, and where we see the bulk of their life cycle is in their haploid stage. So the multicellular individuals are going to produce gametes. They're going to find each other. So we've got gametes. We got fertilization. That fertilization is going to make a zygote. And then with fungi, that zygote is immediately going to go and undergo meiosis. So there's our 4th thing, to make haploid spores, and then the circle is complete. So most of the time, fungi spend their life in a haploid state. Fungi can have thousands of genders depending on their mating types, and depending on how their genes are that determine how their genders are developed. And we don't have the vocabulary to describe more than 2 genders, really, right? Like maybe non-binary, maybe 3 choices, but fungi have thousands and thousands and thousands, and it's all genetically determined based on their types of alleles. Another weird thing that fungi do is they can have, so if you think of fungi, these are these big long strands of cells just stacked end to end to end to end, and sometimes those are hollow and there's not a cross wall in between them, and so it's just a big long tube. You can think of like a straw filled with cell juice and with a bunch of nuclei floating around in them. So that's a weird thing that fungi do. Fungi do a lot of weird things, and they're cool, and everybody should take mycology because it's awesome. All right, reproduction in animals, we have gametic meiosis. So what's going to happen is most animals spend the majority of their life in their diploid stage. So, and then we have specialized cells that undergo meiosis. Boom, there's meiosis. This is going to make gametes. Gametes are going to find each other, do fertilization to make zygotes. So then there we go. We hit all of those things. Okay. But there's always exceptions to the rules. So let's just talk about some of the exceptions to the rules in this case. So some animals, like such as wasps, bees, and some ants, their biological sex is determined on their ploidy, by their ploidy. So female bees are diploid and male bees are haploid. And so male bees develop from unfertilized eggs. So if we look at this, so this is how we come up with male bees. We have our female, she undergoes their cells that undergo meiosis in her body. We get some eggs that will develop, do mitosis and develop into multicellular male individuals. If we have our diploid female bee, and our haploid male bee, there's no meiosis that occurs, right? They just produce sperm. That's a gamete. Our female, our queen, is going to produce eggs through meiosis. They're going to find each other, and now we have two diploid female bees. So this is pretty cool. I like this little exception to the rule. It's pretty neat. And again, we tend to find them in this group, bees, wasps, and ants. And they're cool. They're cool. So we have different kinds of cells. So you guys are going to want to know the difference between somatic cells and germline cells. Somatic cells are what makes up the majority of our bodies. So these are diploid. In animals, most animals, they divide via mitosis. So if you need to replace some skin cells, you're going to do mitosis to replace those cells that you're shedding. And then our germline cells occur only in our gonads. They produce haploid cells via meiosis. And yeah, and I already said that they're in our ovaries or testes, depending on which kind of gonads you have. Okay. So I want you to think a bit about this a little bit and think about why do we see sexual reproduction? It's expensive. So asexual reproduction, you only need one individual to reproduce. But for sexual reproduction, you need two. And like when you try to collaborate with people, it gets complicated. And it's like asexual reproduction is faster, it's easier, it's less costly. So why do we see almost all eukaryotes able to do sexual reproduction? So think about that for a minute. And then also remember, tuck this little image here of the Red Queen talking to Alice from the children's book, Alice Through the Looking Glass. So we're going to keep that in our mind, put that in our back pocket. And I want you to think about, why do we see sexual reproduction? Okay. So now let's talk about meiosis. We're going to get into it. So meiosis has 2 rounds of division. And because we have 2 rounds of them, we're going to call them meiosis 1 and meiosis 2. Each of these has a prophase, a metaphase, anaphase, and teleophase. And we call it prophase 1, metaphase 1, anaphase 1, teleophase 1, and then it goes into prophase 2, metaphase 2, anaphase 2, teleophase 2. All right. So some of the things that are different about mitosis is that the first thing that we see that's different is we see this synapsis. This happens early on, and the very first thing that we're going to do is we're going to pair up our homologous chromosomes. So if we get 1 chromosome from mom, 1 chromosome from dad, we're going to pair those up. They have the same genes. They might have different alleles for those same genes. If you might need to go back and look up what an allele is if you don't remember. But they're going to synapse together. So the way that I remember this is, but the homochromos are going to synapse together. Okay, so homologous chromosomes are pairing up. And that's important because for crossing over, that we'll talk about in a little bit. All right, so here we go. Let's talk about this process. So first of all, we have interphase, right? So our cells are just doing our cell thing. They're just kind of hanging out, doing whatever, right? That we still have growth 1, G1. We have our DNA replication. And then we have our G2 phase, and now we're ready to do meiosis. So first thing in prophase 1, we're going to see the synapses. We're going to have our homologous chromosomes that are going to come together. They're going to do crossing over. I'll talk about more about that in a minute. Then they're going to line up. Here's a thing that's different about mitosis. In metaphase 1, the spindle fibers are attaching to the homologous chromosomes. So I got a spindle fiber here on this homologous chromosome and a spindle fiber here on this homologous chromosome. And what's that's going to do in anaphase, that's going to pull apart our homologous chromosomes. Okay, and then of course we'll have telophase, where we are rebuilding that nuclear envelope. So as soon as we get to prophase 2, we're going to have to take out that nuclear envelope that we just made. We're going to have to deassemble it. Then we see these spindle fibers attached to the sister chromatids, similar to what we saw in mitosis. So when we're here in meiosis 2, or metaphase 2, it's going to pull apart. And now in anaphase 2, it's going to pull apart those sister chromatids. So then we can reach telophase. And now we have those 4 haploid daughter cells. So here in this anaphase 2, that's boom, that's when you become, nope, sorry, anaphase 1 is where you become haploid. Okay. Okay, so meiosis 1 is called the reduction division. The daughter cells that contain 1 homologous chromosome for each from a homologous pair. We don't see any DNA replication between meiosis 1 and meiosis 2. That would defeat the whole purpose of trying to get down to being haploid, right? We copied out the DNA again. So we see a repression of DNA replication between meiosis 1 and 2. Meiosis 2 separates the sister chromatids. That's going to be an important distinction to make. So make sure that you understand the difference between a chromosome and a chromatid. Okay, and then we see that G1, the S phase, and the G2 phase, like I talked about. And okay, I think that's all I'm going to say there. I already talked about all that. Okay, so in prophase 1, boom, number 1, boom, prophase. We see, so in interphase, all of our DNA is just kind of like loosey-goosey floating it out. In order to get ready to replicate, we got to pack it all up, just like you pack up. And when you're ready to move house, you're going to pack up all your nice fine china, and you're going to make sure it's safe. You're going to make sure it's labeled properly. You're going to make sure that it doesn't get lost or broken. And that's what's happening with the DNA. So we're going to coil it up, and we're going to make it into these condensed chromosomes, so everything stays nice and tidy. Okay. We got to get rid of that nuclear envelope, so it's not in the way, because we got to pull, remember, we're going to eventually pull those things apart. So we got to get rid of that nuclear envelope that's going to be in the way. We're going to have our spindle fibers form when we're talking about animal cells, so that can yoink those chromosomes and chromatids apart. At this stage, each chromosome is composed of 2 sister chromatids. So if we look at this, so here, this little red X here is 1 chromosome, and this little blue X is 1 chromosome. Here's a sister chromatid. Here's the other sister chromatid of that red chromosome. Okay. All right. So again, synapsis happens. And so our homologous chromosomes are coming to hang out. So our copy from mom, our copy from dad are coming to hang out together. And we actually see a little bit of a swap. We see this crossing over occurs between non-sister chromatids. So the chromatids on the other guy, the other homologous chromosome. So let's look at this. Crossing over is one of the sources of genetic variation. So I'm going to ask you later, what are the 2 sources of genetic variation? Well, here's number 1, crossing over. This is changing parts of the chromosome with the other homologous chromosome. So we're switching our genetic material. Alleles that you got from your mom might actually end up on your dad's chromosome and vice versa. So this schismata is, is the site of crossing over. And so here's our site of crossing over. So the $10 science word is schismata, where we actually see this little swappy doodle of our genetic material. Okay, so there's our source of one of our sources of genetic variation. Okay, and it's this. So let's pull the red queen out of our pockets. This is the recombination of genetic materials, is why we see so many organisms doing sexual reproduction. This is the advantage. So if we just had a population of clones, if some parasite or pathogen figured out how to get past our genetic defenses, it could wipe us all out. But because we're a little bit slightly different, it might be able to take out a few of the individuals in the population, but it's not going to wipe out everybody. So having that genetic variability allows natural selection to have that variability to play with. And so if we all have the same traits, if the environment changed that we weren't suited to, we would all die. But because we're all a little bit different, there might be some things that work in this new environment. So that's why we see sex, because it's actually really useful in making sure that our genes get passed on. Even if it's not 100% of our genes, it's some of our genes are making it on to the next generation. Okay. Oh, and then I want to say about the red queen. I threw the red queen in there. The red queen hypothesis is really cool in population biology. We talk about this in evolutionary ecology. So if we think about like a deer, okay, so a deer is going to evolve to run faster than a coyote so the coyote doesn't kill it. Okay, but if the coyote can't catch any deer, or, you know, or whatever, pick your favorite prey animal, they're going to die, right? So now they're going to be selected to be able to run faster. And so then the deer are going to be selected to run faster. And then it's just this increasingly arms race. And so, you know, if you think about Alice in Wonderland, and the Red Queen was like, okay, we have to run. And then Alice is running. She's like, wait, we're staying in the same place. And the Red Queen says something like, oh yeah, the faster you run, the more you stay still. And so really what we're seeing is this evolutionary arms race. But the difference between the predator and the prey, or the host and the pathogen, or, you know, pick your favorite evolutionary arms race, the difference between those two organisms remains the same, even though we see this increasing escalation. And it's kind of the same. The Cold War was the same way. Like, oh, I got a bigger nuke. Okay, now I got a bigger nuke. Now I'm going to get a bigger nuke, and I'm going to get a bigger nuke. It's the Red Queen hypothesis. The faster we run, the more we stand still. Sex. Okay. All right. So here again is just another way to show our crossing over. And then this crossing over is going to make it all the way through to our anaphase 2, where we see the sister chromatids pull apart. But we'll get there. Okay, so metaphase. Metaphase 1. We're seeing the microtubules on these spindle fibers attaching to each homologous chromosome. Okay, so when we talked about mitosis, those spindle fibers are attaching to each sister chromatid. But now it's taking that whole chromosome and it's pulling it to the end of the pole. So now we still have our double bit of DNA, still one chromosome, but now they're being pulled. Okay. And the other thing that we need to talk about is that each homologous chromosome are aligned at the metaphase plate side by side. So here's one of our homologous pairs. We see the chromosome on this side, we see a chromosome on this side. We see a chromosome here, we see a chromosome here. Okay, we want to make sure that each of our daughter cells that are going to result from this has the right number of chromosomes. We don't want this cell to have 4 chromosomes and this one to have 2, we want each of them to have 3. And so this lineup is going to ensure that the right chromosomes end up at the right poles, so they end up in the right cells, so that we can go on to do the rest of meiosis and make sure that our resulting cells are going to be viable. All right, so now anaphase. Those microtubules on the spindle fibers are going to shorten, and that's going to, so we're here, so we're going to be pulling them apart to anaphase. The homologous chromosomes are pulled to either end, so that's what we're seeing here. Now, we are now haploid. Now we're seeing that separation of our 2 chromosomes. Okay, now these are doubled, right? Because they have their sister chromatids. That's different from homologous chromosomes. Now, source number 2 of genetic variability comes from independent assortment. When our chromosomes are lined up on metaphase, it's not necessarily all of all the chromosomes that we get from dad are going to be on one side, and all the chromosomes we get from mom are going to be on the other side. It's going to be selected at random. And so all of the sperm cells that you make, all of the egg cells that you make are going to have a different combination of the chromosomes that you got from your mom and dad, resulting in those sperms and eggs, your gametes. And we also had crossing over, and so there's a whole nother level of shuffling of our genes. So if I'm going to ask you what are the 2 sources of genetic variation, you're going to say crossing over and independent assortment. And crossing over happens in prophase 1. Independent assortment happens in anaphase 1. Okay, so now we've got our telophase. We've got our chromosomes all at one of the poles, and so now we're ready to build our nuclear envelopes back around these 2 dudes. Our sister chromatids are no longer identical because we had that crossing over way back in prophase 1. Now, we may or may not see cytokinesis at this point. So remember, cytokinesis is the actual pinching off of the 2 new cells. We have 2 nuclei, and they could be hanging out in one cell, and then go on to do the rest of meiosis 2. Kind of depends. And then meiosis 2 is going to happen at some point. And depending on what kind of gonads you have and environmental conditions and all this stuff. Okay, so meiosis 2 is very much like mitosis. Okay, very much like a meiotic division. We have prophase 1. First thing we got to do is get rid of that nuclear envelope that we just made. It's going to dissolve. We're going to have new spindle fibers develop. Along in metaphase 2, we're going to have the sister chromatids line up, spindle fibers attached to each sister chromatid. It's going to pull those apart in anaphase 2. And then in telophase, our nuclear envelope is going to reform around our haploid chromosome. So we're going to end up with 4 haploid daughter cells that are genetically distinct from each other and from their parent. And I think my battery is going to die, so I'm going to pause this for just a second and plug in my computer. Oh, how do I do that? Okay, crisis averted. So, okay, all right, to wrap up meiosis, our final result is 4 haploid cells. They are genetically distinct from each other and genetically distinct from their parent cell. In us animals, these are going to develop directly into gametes. In plants, fungi, protists, some other critters, they're going to undergo mitosis, yes, I said that correctly, and they're going to create multicellular individuals that will go on to create the gametes. Okay, so there we go. Takeaway of meiosis: sexual reproduction, 4 cells genetically distinct from each other and from their parent. Haploid. Those are the things that you need to know about meiosis. Okay. All right, these four features. So synapsis of homologous chromosomes provides us crossing over way back in prophase 1. Our sister chromatids are going to remain attached to each other at their centromeres. That's just a little part, that's the joining part of the X, where those sister chromatids are attached to each other. And that is going to go all the way through meiosis 1. And when we see anaphase 2, that's when we start to see the sister chromatids being pulled apart. And then our cores of sister chromatids attach at the same poles so that we make sure that we get the right number of chromosomes in the right cells. And we do not see DNA replication between meiosis 1 and 2. Those are the takeaways there. All right. We can have meiosis go wrong. So what's going to happen if meiosis is wrong? So we might see non-disjunction. So under normal meiosis, we see all of our daughter cells having the correct number of chromosomes. If we have a non-disjunction event where we actually see these spindle fibers pull over an extra set of those, both sets of homologous chromosomes, then we're going to see that our cells, our resulting cells, are going to have either too many or too few chromosomes. If we see non-disjunction in meiosis 2, then we see 2 cells that made it through, that did it right, and then we have 1 cell that has too many and 1 cell that has too few. And so we can kind of track that. Typically, if we have gametes that have too many or too few chromosomes, like these guys, if that gamete survives and participates in fertilization, generally that zygote is aborted. So that's spontaneous abortion, is where nature's like, this isn't going to work. Because what do? How do you do, if you have too many or too few chromosomes, your gene expression is not going to be right. And we actually see this in humans that have Down syndrome, for example, where there's an extra 21st chromosome. We see developmental differences in folks with Down syndrome than folks that have just 2 copies of each of their chromosome number 21. For example, I have a friend who has a baby that has trichomy 18, so she has a third 18th chromosome, and she has a lot of developmental issues, a lot of health problems. Okay, so that's the end of meiosis. So let's talk about weird stuff, shall we? I love weird stuff. We're going to do parthenogenesis, or if you're talking about plants, we call it parthenocarpic. So it's just like Genesis is the beginning, and I forget what partheno means. I should look it up. So let's talk about it. So usually in meiosis in humans, we see, depending on what kinds of gonads you have, if you happen to have testes, you're going to start making, your meiosis is going to take a couple days, maybe a week. You're going to start doing that at puberty, and you're going to keep doing that for the whole rest of your life. When we're going to produce sperm, of course, sperm have a head that has all of the stuff in it, and then a flagella that allows the sperm to find the egg. Now, if you happen to have ovaries like me, then you're going to start doing, meiosis begins as a fetus. So even before you're, way before you're born, meiosis starts. And so this can be delayed for months or years. And in humans' case, it's usually years. So our meiosis 1 occurs about 14 weeks of pregnancy. And then meiosis 2 won't occur until ovulation occurs. And then the number of eggs we get that we're born with that's how many we get. We don't get to make more. And then when we run out of them, then that's when menopause happens. All right. So what we see is that even though we usually make 4 gametes, we see that 3 of them actually don't actually make it all the way through, and then all the investment goes into making 1 egg from our 1 cell that underwent. So it's like kind of a 1-for-1 shot instead of a 1 for 4, because 3 of them are wasted. But I mean, let's be honest, guys, you guys waste more gametes than we do. Okay, so let's talk about parthenogenesis. So this is when we see reproduction from an egg. So egg is the fancy, or ovum is the fancy science word for egg, where we see reproduction from an egg without fertilization actually happening. This usually happens from an unfertilized egg, like we talked about with the bees, but we can actually see it in some other kinds of critters. So this is automixis, is where we have fusion of two, usually egg cells derived from the same individual. And so the eggs are fusing to form a new diploid, and that's going to undergo mitosis and develop just like a normal thing, but it's a clone, right? You might have a little bit of genetic variability from crossing over, but not as much as you would from like proper sex. All right, so let's talk about these cool little lizards that do this weird thing. So we have, there's these, so in this genus, which is my goal is to be able to properly pronounce and remember this genus name, because these guys are super cool. But what we see is we have we actually have 3 distinct but closely related species of these little lizards. And what happens is, this guy can go off and mate with members of its own species, and this guy can go off and mate with members of its own species, but they can also mate with each other and form a hybrid that we call this species. And then this diploid organism can actually clone itself, or it could undergo sexual reproduction with another species that's closely related that lives in the same area. And now we see a parthenogenic triploid. And so here we have an organism that has 3 copies of each chromosomes. But how do you split up chromosomes? And if you have 3 sets of chromosomes, how do you split those evenly amongst 2 cells? You can't really do meiosis properly, right? If you have you can't split up an odd number into even whole numbers. You can't do it. And so these guys actually can't reproduce on their own. They have to, what they actually do is these guys can clone themselves, and what they'll do is they'll still interact with each other in kind of the mating way. So they'll like kind of do this little wrestle thing where and that gets their hormones pumping to say, okay, it's time to reproduce, but they're actually, what we see is actual cloning instead of actual sexual reproduction. So this is pretty neat that we have this in such in a vertebrate animal. So that's pretty cool. Another thing that we can do is look at some organisms can both asexually and sexually reproduce. Most plants can sexually and asexually reproduce. And so when we talked about mitosis, I talked about how I go around and take cottonwood cuttings, and I go and knock off a branch, and I take that branch back to the greenhouse, and I cut out little branches, I stick them in rooting hormone, and I grow all of these cottonwoods in a greenhouse environment so that I can go out then and plant them, do an experiment, or I can go off and plant them at a restoration site or whatever. And so that's asexual reproduction. But how plants want to do it, because while cloning is really convenient for me, where I want to study the genes and the environment and how important genes are and how important the environment is to the way that the plants express their traits, that works for them, but they want to make sure that they're going to be able to survive and that genes are going to be able to be passed on. So they want to have sexual reproduction so that maybe their kids are going to be better suited for their environment than they were, right? That's why we do sexual reproduction. So all that long-winded way to say is that I also, when I was an undergrad, I was studying these aphids that form these growths on plants, so that what they do is the aphids will trick the host, the plant, into making them a little bug house. And so what happens is in the early spring, a stem mother will emerge from an egg. So as the leaves are starting to break bud and starting to open up and grow, as that these stem mothers, they will fight to the death, and they'll kill each other, and they'll stab their foot in the other one, and they'll rip its guts out, and so they're really competitive, and they fight really hard for their favorite gall spot is right here at the base of the petiole. If they try to do it up here, it doesn't usually work out so well. What they do is they stick their nose into the leaf's vascular tissue, and they modify the plant's hormones, and they say, hey, make me a little bug house. And the plants are like, okay, yeah, we'll make you a bug house. All right, it sounds good. And so we see this folding up strangely of the leaf, and then it grows into this little gall here. And inside that gall, we actually see that stem mother making clones of herself. And so all summer long, all spring, all summer, she's in there cloning herself. And so these galls can have 50 to 300 new individuals that are clones of the stem mother. And so in the early fall, will break open, all of those aphids will fly out, and they'll go feed on the roots of another plant that rumex, it's a weed that these bugs eat. So they'll stay on that plant until winter, and then in the winter, right before, like late fall, early winter, they'll fly back to the cottonwood trees. They'll mate with each other and have proper sexual reproduction. The females lay the eggs, and then those eggs overwinter on the trees, and then we go back to doing this weird gall making thing. And so, okay, why? Here's a great essay question. Why might you have the ability to, why are you going to have asexual reproduction? We already talked about why sexual reproduction is so great. What's so great about asexual reproduction? Well, first of all, it's cheaper. You only need one person. It's easier. And inside of this gall, do you think that is going to be a stable environment, or is that going to vary a lot? It's going to be pretty stable. It's going to be pretty consistent inside of that. And so if you have the genes that says, hey, plant, I'm going to eat the sugar that you make through photosynthesis, and you're going to keep me safe. And the plant's like, okay, yeah, that's a good idea. Then, like, that's a good strategy. You want to keep those genes. You don't want to tweak that. You want to keep that the same. But when it comes to be wintertime, you don't know what next year is going to be like. You don't know what tree you're going to fly to. So then you want to have that genetic mixing, right? And so this is a good essay question. Under what conditions is asexual reproduction the most advantageous? And under what conditions is sexual reproduction advantageous? Why would you do sex? Why would you do asex? All right, essay question. All right, this is one of those neat, nice to know things. This is a nice summary slide of all the different ways that we might see parthenogenesis or parthenocarpic reproduction. Like dandelions, they do this apomixis thing where when you pick a dandelion head and it's got all this little fluffiness, and you go, and all the dandelion seeds go flying off, they were actually produced from a diploid egg. And so they're actually clones of the plant that you just blew. So dandelions like to be clonal. All right. And then this last little bit, I'm not going to test you on this, so I will just stop the lecture here. I think I've rambled for a long time. So definitely check out these slides. But I'm not going to, these ones won't appear on the exam. Okay, let me know if you have any questions, and I will see you later.